Yeah. Please tell me that I can't, that I won't, that I fail, that I'll never make it out, yeah. Please tell me all the bad, never good, fill my head full of every single doubt, yeah.
Okay, hello, I'm Tim Sandle and I'm a Pharmig committee member, and it's my great pleasure to be speaking with the Pharmig chairman, David Keane, and we're going to be looking at a number of areas of disinfection, contamination control, and other hot topics of interest to pharmaceutical microbiologists. So welcome, David.
Thank you.
And just to start with, please can you tell the audience a little bit about your job with Ecolab and what it entails?
Okay, yeah, so I've been with Ecolab about 6.5 years now. And my role is essentially we work with our customers, sometimes even not our customers, as microbiologists. Ideally, we work with them on issues we can help them with from an Ecolab point of view, but quite often I've been working on complex microbiological issues, and everyone seems to appreciate the effort. So it's microbiology with our pharmaceutical customers, and it's quite a varied life. We'll be looking at disinfection issues one day, water systems the next, to product failures on the third day. And my role is global, so I've spent a lot of time in Asia Pac recently.
Oh, right. Okay. That sounds very globetrotting. Yeah. Okay. So kind of moving on to the main questions. There's been a kind of real buzz since Annex 1 came out, the revision in 2022, about the contamination control strategy. And I was just wondering what the CCS concept means to you.
Well, apart from its meaning, contamination control, which as a microbiologist is it's close to my heart, I guess for a lot of people it's same old. It's just the quality management system linked to risk assessments and with a bit of plan, do, check, and act, and a feedback loop that means that when we get some results which indicate fix them, but then we strengthen our controls to prevent it happening again. The contamination isn't just microbiology. Obviously, there's chemical and physical. If you're not familiar with this before, I've recently dealt with a customer who their belief of the particulate contamination was only based on airborne particulates in clean rooms. So there's still quite a bit of understanding that needs to be breached on that. So it's all forms of contamination, physical, chemical, and biological. But obviously, with my background, there's a strong basis on the microcontrols.
Yeah, linking to that, how does cleaning and disinfection kind of connect into this sort of CCS concept, and what should people be including relating to that area?
Well, from a micro point of view, it's one of the major control points for the contamination point for in the clean rooms itself and ancillary areas. And the main reason for that is that there's a huge number of controls that are built into our clean rooms, from our HEPA systems, our pressure cascades, to the room air exchange rates, for example. But then we put people in the room. And as you know, people are the biggest source of microcontamination. So we need some way of cleaning up their mess, as we say. And disinfection is the main means of doing that. And we need cleaning because disinfection doesn't work unless it's applied to a clean surface. So the two go together, but two distinct separate processes. And obviously the cleaning process will also aid with chemical and physical contamination risks as well.
Particularly the emphasis on cleaning, which you're right, is something people tend to overlook. So with cleaning and disinfection, one of the kind of endpoints is the environmental monitoring data. So what can people infer from their EM results that might lead to how they view their cleaning and disinfection?
Yeah, so the two go like hand in glove. If you've got a cleaning and disinfection program, you need an environmental monitoring program, and vice versa. If you've got environmental monitoring and you get some results you don't like, you need cleaning and disinfection, principally disinfection. So you can use your environmental monitoring program to demonstrate your disinfection process works. Very basically, if you monitor before you disinfect and then you monitor after you disinfect, you'd expect the results to be better because you've destroyed any micro contamination that might be there. In terms of the impact cleaning and disinfection can have, then we're reacting in terms of environmental monitoring to what we find. Now, it's always in the past. Even with the more rapid methods that we get there is still sometimes a delay of several days. And we need to use our environmental monitoring as a trending tool. So we should never be using EM as each individual data point. It's not as valuable as an overall trend over time. So the EM trend tells you about the general health of your facility, and you would be expecting to see your routine organisms that you find time after time, which you know your disinfection program can cope with. And then you're on the lookout with your EM program for those organisms that might indicate a loss of control. So environmental spore formers such as bacillus are a good example, and that might be a problem with material transfer, for example. And this is where you will be able to potentially react with the use of a sporicide or other more aggressive control points. And then something like molds appearing in a clean room sometimes is indicative of changes to your air patterns in your clean rooms as well. So the EM tells you something about the state of the health of your clean rooms and what the people in there are doing. And your disinfection program, it's reactive to it, but ideally, in the best world, it's also a really good preventative if we get our rotations correct.
Okay, that's good. Actually, that thing on rotation was actually my next question. And we spoke earlier about sometimes needing to go back to basics. So I think there's still sometimes a little bit of confusion about the need for disinfection rotation and how often to rotate and what to rotate with. So just wondered if you could share your thoughts on that, please.
Yeah, so the regulatory guidance out there isn't brilliant. There's no black and white, you must do this. USP 1072, United States Pharmacopoeia, has really good guidance for cleaning and disinfection, and it talks about what rotation is. There's some good stuff in PICS as well. Udrilex Annex 1 for sterile manufacturing. It's good, but in my opinion, it could be a bit better. So from a scientific point of view, rotation is the requirement to make sure that you have a disinfectant regime that can kill everything that you would routinely encounter in your clean room environment. And one of the reasons that we do the rotation, and rotation is with a broad spectrum disinfectant that can kill vegetative cells, and then you would normally rotate it with a sporicide, so something that's able to destroy bacterial endospores. And to be clear again for the basics, we're talking about bacillus endospores and not Clostridium endospores. Sometimes people with a healthcare background will focus on Clostridium for very valid reasons, but it's not as resistant as bacillus. So we focus on bacillus. Now, if we were just using a sporicide, all sporicides are oxidizers, strong oxidizers, it will kill all vegetative cells as well. But the issue with these sporicides is they're really aggressive. So they can cause damage to your facilities, or sometimes there are some safety exposure limits with some of them. So by rotating a broad spectrum with your sporicide, you're able to cover your clean room in a way that minimizes the risk against contamination occurring, but also potentially reduces the risk of damage or corrosion from these more aggressive chemicals.
Trying to assess the frequency of sporicide to non-sporicide, is there a good approach to take?
There's definitely a bad approach. A long time ago, I was given an observation for having a reactive sporicidal approach. So we only used a broad spectrum disinfectant, and then when we recovered spore formers from our EM, we would then go in with a mop and a sporicide. But as an inspector gently pointed out to me that we're always working in the past, the contamination has the potential to have got worse before we react. So a preventative measure, and obviously this is in line with your CCS, is to rotate with your sporicide. Now, generally, the rotation frequency is up to the site. There's no real guidance that helps, but you need to use your environmental monitoring program and also a risk-based approach. So if you've got a more critical area, if you've got your ISO 5 areas or your grade A and B clean rooms, you would expect to do the rotation with a sporicide much more frequently than you would with a grade D or a grade C clean room, because the risks in those areas are lower from these organisms than they are in our more sterile environments. So we use our EM as a baseline in exercise, 1, to define our frequency, and then 2, to ensure that the frequency is robust. If you've got a new facility or a new line, you're not going to have that baseline data. So again, speak to your disinfectant suppliers. They give you really good guidance on what to look at. But essentially, the further you are away from a sterile core, then the less frequently that you will need to do your rotation.
Okay, no, that's really useful. Something that comes up at conferences from time to time is questions relating to disinfectant efficacy testing. And there's obviously the emphasis that the surface test is a key test. If a facility has lots and lots of different surface materials, maybe over 10, 20 different types of materials, and given like the costs of disinfectant efficacy testing, how do they sort of narrow down the number of surfaces, or would they have to still do every surface, or how do they deal with that conundrum?
So again, throughout this podcast, I suspect if you did a count, I'm going to use the word risk a lot. But it's the current nature of the world that we're in with pharmaceuticals. A risk-based approach is what you would look at. You could test every surface in your facility, but you'd have to question, does it have that much value if you've got some surfaces that are only a very small percentage of your facility and are perhaps remote from where you're actually doing some critical work? There's also, as you've alluded to, a significant cost to doing these additional surfaces. So the least risky option would be to test everything, but you have to balance that against the value that it gives. So we often look at a matrix approach. We will look at the number of cleanroom surfaces that there are in terms of number, but then also in terms of type. And one of the ways that you can reduce the number of cleanroom surfaces that you test is to look at sort of the similarities of material. So a good example of material similarities would be stainless steel. You've got a standard material is stainless steel 316L, and then you get a more mild steel 304, sometimes seen. Now 304 stainless steel is rougher generally than 316. And both the materials are the same. So if you were to test on 304 stainless steel, you could define that as your worst case material, and then you could eliminate the need to test 316. And you could do that with similar materials like glass and polycarbonate, for example, from like an isolator, a wall or a viewing panel, for example. Other things that we look at will be the hydrophobicity of the material. So items that are very hydrophobic, what those items do when you put a disinfectant on, because all disinfectants, the solvent used in those disinfectants is water. If you add the disinfectant to a hydrophobic surface, then you get more of a complete ball on the surface, the droplet, and that means that the surface area of the disinfectant touching the coupon is much lower. So if you've got materials that are similar but have different hydrophobic properties, again, your worst case risk will be the more hydrophobic one. And then another risk factor to look at will be surface smoothness. I touched on it briefly with stainless steel, but if you've got a cleanroom surface that's rougher than another one, then there are more places for microorganisms to adhere to, the surface area is greater, and it presents a greater challenge to the disinfection process, trying to get those molecules of the chemical to the microbial cells. So 3 things: material type, similarity, how hydrophobic it is, already forgotten my last one, surface roughness, and then the final one will be the percentages in the materials of the present and criticality. So if you've got a very, very critical material, but a small percentage, you might want to cover it. But if you've got a material of low risk, low location, you might want to discount that anyway, because the value of testing it is not outweighed by the risk it would bring. It's a big subject, and there's a lot of help that can be given, but that's roughly how you would do it.
Yeah, no, that's really excellent advice.
Thank you.
There's another question. So some facilities would periodically use a gaseous or vapor disinfection, something like hydrogen peroxide. Is there a role for that in the cleaning and disinfection strategy? Is that something that's common and necessary?
It is common, and the word necessary depends on your process. So I've worked in facilities that use it, and I've worked in facilities that don't, and they remain a completely manual disinfection process. So the benefits of it, so if you're looking at something like hydrogen peroxide, then it's a sporicide. And when you have an automated system, which, these gassing systems are all automated, then it's very reproducible and it's very robust. So when you compare that against the manual application of a sporicide, then the automated system is going to be better in terms of you're going to get better distribution. Once you've done the validation, you're going to get a more reproducible kill. Because when we give people a mop, despite the level of training and education, all those human error factors can come into play, and we might make an error, we might miss a bit. And then, because of the way the microbiology works, that's where the contamination will be. So you have this advantage over manual disinfection, but there is a disadvantage in that it can take significantly longer periods of time. So if you're looking to use this as part of your rotation, a good example where we see this used is people will use manual broad spectrum disinfection during routine operations, and then overnight they might use these systems when the clean rooms aren't in use. But obviously, if you've got a 24/7 facility, then that might not be appropriate, because the amount of downtime that you have here to deliver this disinfection process might outweigh the efficiencies of using the manual process. So it's about finding that balance. Another really good place that we see these systems being used for clean rooms is after a stoppage or things like a planned annual shutdown, where the rooms are often declassified or lowered several grades. You get lots of contractors coming in, some not necessarily with the greatest of clean room knowledge, and we're bringing in lots of equipment or calibration equipment, engineering machinery to help construct something, all of which will potentially add to a much greater microbial challenge. So a lot of companies will use these systems as we come out of the shutdown to give a very heavy duty hit on the clean rooms, and then it allows the bioburden to be reduced to such a point that the manual disinfection process can then take over. Another good example would be customers who are perhaps working with viral vectors, and you might look at something like a HIV virus. You might want to, as a protection point of view, gas clean rooms because of the ability to be virucidal as well. So there is a place for it. You just have to work out what the benefits are versus the downsides, because there, in the disinfectant world, there is no perfect disinfectant. Everything has its good points and its bad points, and we just need to find that balance.
Okay, no, that's really good advice, and can see how people could implement or not, as the case may be, in their programs. This is more of kind of like an open question, but from your sort of travels around the world, are there any particular horror stories or weaknesses with contamination or cleaning and disinfection that you can share with the audience?
Well, you generally, when you go somewhere, you will always see something. Before I joined Ecolab, I've been to a lot of places around the world as well with various different companies that I've worked for, and there are always some interesting things to see. I've seen people climbing into transfer hatches to perform the disinfection process, not particularly gowned very well. I had the delight of working in a facility, and I couldn't understand why my environmental monitoring trends were so high for grade C. It just didn't make any sense when I looked at the number of recoveries and what we were doing in there. And then when we actually reviewed how we were doing the disinfection process, we were adding disinfectants to hot water, which were deactivating them. So again, what we were disinfecting the floors with was not really a disinfectant. It was really just a sticky water. I've also seen, again, at a facility I worked at, where the operators didn't like the smell of the disinfectants, and so they were adding less, so it didn't smell as bad. And obviously when we have a lower dose in there, we're not getting the level of kill that we need. I mean, I could go on this topic for hours, but the incorrect types of mops, the incorrect types of cleaning equipment, where we see all sorts of secondary contamination caused by these, and most of it is just people don't understand. Once you explain to them, then these errors and mistakes just vanish. So it's just an education piece. And I think cleaning and disinfection, certainly when I was in industry, it's just something that everybody does. And if you join a new company, or if you join an existing company, somebody put that in place before you joined. So it's always been there. And by and large, your environmental monitoring, which we spoke about earlier, says it's okay. And most microbiologists working on the site have got bigger fires to put out than cleaning and disinfection. So these little things tend to run in the background unnoticed until they have time to spend some time and then go, oh dear, perhaps we can improve it this way.
Yeah, and there's that whole thing about knowledge transfer, and if you're in the industry long enough, everything just repeats around.
It does, yes.
Yeah. There's been discussions around residues and residue removal, and this was something that in one of the earlier drafts of Annex 1 it was given greater prominence, but so it's still there, but not as much. But is this still an important subject? Is it something that regulators are looking at, and what approaches should companies be taking if they have concerns about either detergent or disinfectant residues?
Yeah, so again, Annex 1 was updated for three reasons. One, to improve the language for people who don't have English as a first language, to bring it up to speed with changes in technology, things like isolate technology, and also then to bring into line with other regulations such as US FDA. So the call out for rotation for residues has been there for a while. The main residues risk normally is associated with product residues. So from cleaner validation, the need to not cross contamination from different product types on filling lines, etc. When I started in industry, disinfectant residues were often seen as evidence that the disinfection process had been performed. And so people were quite proud to point out the streaks on windows and things, going, look what we have done, this is evidence. But if you look at your CCS and what the CCS is trying to control, which is contamination, physical, chemical, and micro, then these disinfectant residues form both physical and chemical risks to your process. So the residues, they are a potential risk. The closer you get to your critical operations, then the risk of transferring these residues into your process is higher. So understanding what those residues do and don't do is important. Now, the residues are a natural process of adding these disinfectants to these surfaces. Not all disinfectants have the same residue profile. So I think most people are familiar that alcohols and things like hydrogen peroxide, they're volatile, so they evaporate. Or in peroxide's case, it breaks down to oxygen and water. So it doesn't leave any residues as such. But other disinfectants, especially a lot of the sporicides such as peracetic acid or chlorine, they will leave quite a heavy duty residue. So now you've got to find that balance between, I need to use a sporicide, but I'm potentially creating a different risk. And again, it comes full circle. This is why we need cleaning. We clean to remove those residues so that the disinfection can work. So there's a lot of work that goes into how we manage it. In terms of what the regulators are doing, then it is now in Annex 1. It is now called out as a specific risk. You know, the risk from disinfectant residues need to be managed. I think I'm still waiting to hear on some definitive observations directly associated with a disinfectant residue and transfer into production, because again, they are a risk, but how you get it into your process is often quite convoluted. So it can be deemed quite a low risk, but they do cause a problem. They're also, from a health and safety side, they can be quite a risk. When I started, it was quite common to have sticky floors, yellow residues on floors. But we have seen some quite nasty injuries from slips and trips caused by these residues on things like clean room floors, especially when you combine them with the ubiquitous blue clean room overshoe. You're essentially creating a skating rink in some places. So there are lots of reasons to control these residues. And Annex 1 is simply being brought up to date to say it's one of the more pressing things that need to be managed.
Other than the obvious, is my floor sticky? Is there any kind of testing that can be done to try and show whether residues are present to a certain level?
Yeah, so there's lots of things that you can do. What I would say is please don't go down the route of cleaning validation, because the manual disinfection process is exactly that. It's manual. So if you apply this onto a surface, different people, different strengths, even different heights, they're going to get potentially different levels of disinfectant on the surfaces. Now, we all want a minimum level to be able to give us that kill. But if someone's got even something like a pre-saturated wipe, the weight of my hand on the surface will be much different to somebody else's. And that can give you a different level of volume of liquid that leaves your wipe and goes onto the surface. So you can't use clean and validation because you're going to see too much variation in your results. There's lots of chemical methods to look at disinfectant residues. And the main reason for that is that the disinfectants are dissolved in water, but then you will have either a volatile, which will evaporate, or you will have a solid, which will be left behind, a precipitate that comes out, which is your residues. So we, as Ecolab, we've done a lot of work where we've used conductivity. Not everything in a disinfectant will cause a conductivity change, but every disinfectant does have a conductivity level. And so there's a direct correlation between if you took a rinse off a clean room surface and applied the conductivity meter to it, and you compare it against a blank, you've then got something that you can look at and says, this is the difference when I add these residues. And it's important because if we're trying to remove residues from a surface, because we can't do cleaning validation, you're looking at a visual standard. So if you can get something visually clean, you can use that to say, well, actually, visually clean means this with the human eye, but here's our residue profile with conductivity. Now, there's other things you can use in the labs. You've got HPLC. I'm sure there's lots of other chemistry things. I'm a microbiologist, so my knowledge is fairly limited. But we've chosen conductivity as a supplier to industry because it's mobile. It's cheap, relatively, to have a conductivity meter calibrated, and you can bring it into the clean room rather than having to bring samples out of the clean room into the lab. So it's a more rapid process as well.
Okay, so my final question to finish with is that obviously you're not just a global contamination control expert, you're also chairman of Pharmig, and there will be some people watching this video, because we're going to put it out across social media, who are either not members of Pharmig or may have not even heard of Pharmig. So I just wondered if you could just spell out what Pharmig is for and some of the benefits should people wish to join.
Okay. Well, so Pharmig, it started over 30 years ago. It's the Pharmaceutical Microbiology Interest Group, but Pharmig is just easier to say. And we are a hub of microbiologists. What we found when Pharmig, well, not when I found, but when Pharmig originally started, it's because the pharmaceutical industry there's a lot of secrets in pharma. There's a lot of patent protection. There's a lot of racing to get a product to market in order to return a profit for all that R&D investment that you put in. And that kind of culture of not talking drives itself down into production and into microbiology. But we all do the same thing. We all do a sterility test. We all do endotoxins. We do the MLT test. We all do EM. And we all see the same failures. So Pharmig was set up to get microbiologists and people who are interested in microbiology into a room and to talk shop and to swap tips and hints on how to improve or how to eliminate or reduce the risk of contamination occurring. So we started off in the United Kingdom, but we're now global. We've got conferences which we run in various countries. We've got the next big conference will be the United Kingdom annual conference in the middle of November. And then after that, in a few weeks' time, well, you and I will be on a plane to India, and we're going to do a conference series in India as well. So we try and move conferences around the world, and we'll try and bring industry experts to the conferences so people can join us. We publish lots of material, often endorsed by our regulators, on all things microbiology, from non-sterile microbiology all the way through to sterility testing. So we've got a whole load of documents and data that can do that. And we've got a lot of training as well. We've trained regulators and we train people. And we've got a lot of online modules which people can find really useful. In cleaning and disinfection is one of them. And we have a committee of experts in microbiology, both from within the industry and as suppliers as well. And the committee is really good at helping. So we often get a lot of queries that come through to Pharmig, and then the committee are all volunteers. They give up the time to help our members solve some of these more gritty problems as well. And we work closely with regulators as well. So we did have a lot of input into the draft Annex 1. So if you're not a member, you should be a member, because you get to talk to experts in the field, not necessarily me, unless you want to be bored to death about disinfectant. But you get access to a whole heap of people who can help. And it doesn't have to be just the committee. If you come to one of our events, you will meet people with a similar interest and will have gone through potentially the problems that you might be about to encounter, and then they can help. So it's great for networking as well. So you should really join. And if you do join, then Tim will give away lots of discounts, often at very little notice. So it's hugely beneficial if you're a microbiologist.
Yeah. Okay, that's brilliant. And so thanks very much for taking the time out. There's been some really excellent advice for developing the CCS, particularly, the rationale for surface selection and the issues around residues, I think, are really pertinent points that some people are grappling with at the moment. So, David, thanks very much for the time. And for people listening, thank you very much for listening. This is a podcast brought to you by Pharmig, as David has mentioned, and please check out the Pharmig website. Thank you.
Thank you very much.